Well, our next speaker is Dr. Bob Delvin from NHURL at USCPA.
Thank you. I don't think I have to convince many people in this room that the standard approach of doing risk assessment based on in vivo toxicology with either animals or humans is really not where the future is going to go. Whether we use toxicity testing pathways or some of the other approaches that have been mentioned, I think at some point in vitro toxicology is going to have to play a major role in risk assessment by EPA and other agencies. So my task today is to address the issue of what is required in order for agencies like EPA to take in vitro data seriously and use it in a risk assessment process. How can it be validated so that INSEA and other groups actually have confidence that what they get from Tox 21 or ToxCast are usable in a risk assessment paradigm? So the goals of the project that we've been working on for the last year or so is to determine how well in vitro exposure models predict in vivo responses using ozone as a model pollutant. And I'll tell you in a few minutes why we chose ozone as a model pollutant. And to do this, we basically are characterizing toxicity pathways in human lung cells exposed to ozone in vitro, characterizing toxicity pathways and downstream pathophysiological responses in human volunteers exposed to ozone in vivo, and developing models, mathematical models that can assess how accurately the in vitro pathways predict human responses. Now, full disclosure, I'm not a modeler, and I didn't stay at a Holiday Inn Express last night, so I have no hope of answering any of your questions about modeling. But Isla is over here in the audience, and actually, David Miller is one of the modelers in our group, and if I get any questions on modeling, I will punt them immediately to David, who will answer them for you. Well, we've done this before. Lots of people have taken approaches asking how well do in vitro method approaches mimic in vivo responses. We just haven't had the tools that we have now to really ask it at the level of detail. But this is an example of some work we did about a decade ago in which we took different size fractions of ambient air pollution particles, and we asked the question, is there a difference in response between the different size fractions, and the coarse, fine, ultrafine representing the fractions. And when we took airway epithelial cells from humans and exposed them to these three fractions and looked at an indicator of inflammation, interleukin-8, we determined that the coarse particles seemed to be more potent in inducing inflammation than the fine or ultrafine particles. And that was true whether we looked at both the RNA level or the protein level. But we took the same particles and we instilled them into mice and looked at downstream inflammation events, for example, induction of neutrophils into the lung, and asked the question, did what we learned in vitro have any relevance to what these mice were doing in vivo? And the answer was it did. Even in mice, the coarse PM seemed to be more potent than the fine or ultrafine particles in inducing inflammation, is measured either by the influx of neutrophils or production of another pro-inflammatory cytokine, TNF-alpha. So this type of approach has been used quite a bit. And in fact, more recently, we've used microarray technology to ask the question, is there a difference in the gene expression in these cells exposed to coarse, fine, ultrafine? And you can clearly see that there is. So our approach has been really to take this type of omics technology and to begin to identify toxicity pathways induced in vitro and ask the question, how well do they predict toxicity pathways and downstream events that are done in vivo? Why ozone? Well, there's more than 30 years of controlled human exposure studies with ozone. There's literally hundreds of papers that have been published describing the effects of ozone in humans. There's more than 30 years of animal toxicology studies. So we've got a huge in vivo database with which to compare in vivo results. In vitro, interestingly enough, there's not that much, particularly with the modern tools, and that's what we spent a lot of our time doing over the last year or two, is doing a lot of the in vitro toxicology using omics approaches, proteomics approaches, bioinformatics approaches. What I'm going to focus on are two aspects of ozone. This would be a side view of the lung with the airway shown in dark blue above. Then you have this layer of epithelial lining fluid in pale green, and you have the epithelial cells at the bottom. and underneath them you have a basement membrane and then circulating blood cells. And the primary hallmark of ozone exposure is induction of inflammation. And we're going to look at two things. One is the production of pro-inflammatory mediators. And one of those pro-inflammatory mediators, interleukin-8, is also responsible for bringing neutrophils out of the blood into the lung. So we're basically going to ask, can we find things in vitro that would sort of speak to this process in vivo, which we know happens. And the experimental design is to expose people in vivo to ozone, 0.3 ppm of ozone for 2 hours. We then do bronchoscopy at either 1 or 24 hours following ozone exposure, and we get two things out of that bronchoscopy. One, we get cells that we can measure inflammation. We can measure neutrophil influx. We can measure inflammatory cytokine. More importantly, we remove epithelial cells from the airways. By brush biopsy, we isolate RNA, and we look at microarray is a measure of looking at toxicity pathways induced in vivo. From the same person that we obtain the epithelial cells when they're exposed to air, we also take those cells and we grow them in culture. So we don't have to worry about inter-individual variations. We're comparing within the same person their in vitro response and their in vivo response. And we do the same thing. This time we expose them to a range of ozone concentrations and a range of time points, but we basically are also using microarray to look at toxicity pathways. How do we know that the dose of ozone that we're delivering to the cells in vitro has anything to do with what those cells see in vivo? To do that, we take advantage of the fact that we can make ozone using the heavy oxygen isotope O18. When ozone reacts with the tissue, it deposits that heavy isotope tag. And by measuring the O18 present on the cells following in vivo exposure and on in vitro exposure, we can find the in vitro dose that exactly mimics what those cells see following the in vivo exposure. And this is just a couple of examples of the study. I don't have time to really show you all the study in detail, but this is looking at effective increasing dose on the cellular response following in vitro exposure. As you can see, as the ozone dose goes up, so do the number of probes that go up. A lot of them are increases, some of them are decreases. And in fact, if you look at them, we see that there's about 29 genes whose expression is changed following 0.5 ppm of ozone. That goes up to 83 following 0.75, up to 162 at 1 ppm. And 29 of them happen to be in common. And in fact, all 29 expressed at 0.5 ppm are also expressed at the higher concentrations. But as you get to the higher concentrations, there are unique genes that are expressed only in those concentrations, especially 1 ppm where you have 101 genes that are actually uniquely expressed by cells exposed to that concentration. That would suggest that different pathways are likely activated depending on the concentration of ozone that's used. And in fact, when we look at some of the pathways, and I'm not showing you all of them, but I've got a few here to show you, we can see that all three concentrations typically are activating the MAP kinase signal-making pathway. This is the pathway upstream that leads to activation of NF-kappa B and inflammatory mediators. We see that all three concentrations also are causing inflammatory response pathways to be activated. But as we get to the higher concentrations, we also see a transition now to stress response and apoptosis, which doesn't really seem to be happening at the lower concentrations. At the lower concentrations, we're also seeing induction of phase 2 enzymes, which are antioxidant defenses. So at the low concentrations, the cell seems to be able to raise defenses that protects itself. As you go up with the ozone, we see more of an inflammatory reaction, and then we get to the highest concentrations, we're seeing more stress apoptosis. So again, this is just an indication that the dose really does matter. Different pathways are likely going to be activated depending on the dose that's used. What about the time? Well, the same thing happens depending on the time after exposure. We can see 5 different clusters. Some genes are activated very quickly. If you look at each of the panels on the left, that represents the number of genes that follow a specific pathway, and then on the right side you see a single line that represents the average of those genes. Some genes are activated very quickly, within 1 to 2 hours after exposure, and by the time you get to 24 hours, they're basically gone. Other genes in the second group of panels there really hit their peak about 4 hours, and then they're gone. Still other genes don't even start being regulated until about sometime between 4 and 24 hours. These are genes that are upregulated. If you look at genes that are downregulated, you see the same thing. So basically, the dose and the time matter. You're going to have different pathways activated with different doses, and you're going to have different pathways activated at different times following exposure. One of the key pathways that we've looked at is NF-kappa B. Everything in pink you see here represents a gene whose expression was increased following ozone exposure. So you can see that virtually all of the components of this pathway were actually upregulated following ozone exposure. One of the downstream events of this, circled in red there, is interleukin-8, which, as I mentioned earlier, is one of the key inflammatory cytokines responsible for bringing neutrophils into the lung and causing inflammation. So we've done a lot of, there are other pathways obviously besides this one, but this is one I wanted to show you because it was so clear that we can really look at individual pathways and start seeing evidence of pathways being activated. And if we take a closer look at this pathway, now not looking at microarray, which is basically semi-quantitative at best, but looking at RT-PCR now, and look at genes involved in activation, NF-kappa B responsive genes. We did 5 different experiments, which are listed in 1 through 5, and then the average of those 5 experiments are shown on the left side of each of those columns. The brighter red they are, the higher induction in that gene. The more blue it is, the more the gene was downregulated. And what you can see is not only NF-kappa B genes, but also genes activated by NF-kappa B pathways are all upregulated in this particular experiment at 1 ppm immediately following exposure. If you look at the time point, however, you can see that this is a very fast event. Pretty much everything is happening within an hour after exposure. By the time you get to 4 hours and 24 hours after exposure, this pathway is essentially no longer active. So again, this whole inflammatory pathway is a rapidly induced pathway. We have other pathways which are actually induced more slowly, some of them not even induced until about 24 hours after. So again, depending on the pathway you're looking at, the time course will really vary. So to summarize the in vitro studies that I've shown you, exposure to the human bronchial cells causes alterations in gene transcription. The inflammatory pathways seem to be up within a few hours. Some of the other pathways, apoptosis in particular, are not up till quite a bit later. In vivo, if we were having this talk in August, I'd be able to show you all of the in vivo data. We finished collecting it. We're in the midst of analyzing it now. And we'll be giving it to our modelers probably in July, where they can actually ask the question mathematically, how well do these pathways I just showed you in vitro predict the in vivo pathways? But they had to have this in June, not August, so I don't have that data for you. I want to change gears and talk about a couple of other things now. One is some of the lessons learned from doing this, some of the opportunities that I think we have. And one of the lessons we've really learned is it does matter what cell you use. We've used a lot of different cells. We've used cell lines, we've used transformed cells, immortalized cells, and in our experience, human primary cells really are the gold standard that seem to give us the best correlation between in vivo and in vitro. No animal-to-human extrapolation needed. Primary cells in our hands, again, respond more realistically than immortalized or transformed cells. And one of the advantages of using them is you can look at genetic, epigenetic, disease, age, and other susceptibility factors with these cells, and I'll give you some specific examples in a few minutes of that. We've compared probably the most commonly used immortalized human airway epithelial cell line, the Bs to B cells, with primary cells, and I can tell you the results are very different. For one thing, the B cells only have 1/5 of the intracellular antioxidant defense mechanisms that a primary cell has. So right away, they're going to respond to an oxidant pollutant like ozone in a very different manner than a primary cell. Most of the in vitro experiments, whether they're using primary cells or cell lines, particularly with the high throughput technology, are using cells that are submerged in medium, in plastic, which is what this looks like, and these cells have this nice cobblestone appearance. On the other hand, that's not what the cells look like in the lung. In fact, in the lung, these cells don't have much liquid on top of them. They have about a 5-micron layer, thick layer of a mucus-containing substance on top. They like to be in an air-liquid interface. And in fact, it's possible to grow these cells in an air-liquid interface using things like transwell inserts that are commercially available. It also conveniently facilitates exposure to gaseous compounds like ozone, volatile organics, because you don't have liquid on top. Well, when one grows these cells at an air-liquid interface, they begin to develop cilia. They begin to make mucin, two of the hallmarks of what epithelial cells do in the lung. They develop more of a columnar shape. They have vectorial secretion. And in fact, if you took a tissue culture section of these and compared with what you got from a biopsy of the lung, they would look pretty much indistinguishable. So we think these cells are actually going to be a much better model. And in fact, if one looks at microarray expression during this process of differentiation, day 0 is the day they're first put on the air-liquid interface, and they differentiate for up to 28 days. You can see that there is a big difference in expression between these genes. If you look at something that's 21 days old, it is not the same cell as a cell that was first put on the air-liquid interface. Some gene expression changes are over 1,000-fold different during this process, many of them over 100-fold. And this is not a transient event. Once these things happen, they stay pretty stable. So as you can expect, the ability of this cell to respond to toxicants, the kinds of pathways that are going to be activated are probably very different in that cell which has been differentiated for 21 days compared to a cell that's basically grown on plastic submerged in liquid. So again, it really does matter what type of cell you use for these experiments. The other advantage we think that for using these primary cells from humans is that one can look at susceptibility factors. At the end of the day, toxicology is an issue of susceptibility. Most toxicants may not impact normal, healthy individuals. They may impact susceptible individuals, whether that's susceptibility because of genetics, because of age, because of life stage, because of disease. And so if we can build in susceptibility with our in vitro assays, we think that would be a much better approach to really providing the risk assessors information they need. This is data taken from studies by Bill McDonald almost 20 years ago, in which he brought in a couple hundred people, exposed them to ozone, and measured lung function. And that yellow line represents the average change in lung function for that population as a whole. That red smudge represents the range of response of those individuals, showing that some of those individuals actually had a 10-fold higher response than others. The blue smudge represents the range on the low side. So clearly, there is a huge range of response, and this is just in young, healthy individuals, not even talking about people who might be susceptible because of age or disease. If these people are brought back a year later, they retain their hierarchy on this scale, implying that there's some long-lived, perhaps genetic factor that may be driving this susceptibility. Back when Bill did these studies, we really didn't have the tools in the 1980s to address this issue. Now we do. And in fact, it's now known that there's a number of genetic polymorphism that have been shown to be associated with susceptibility to air pollution. One of the more common is glutathione transferase M1, which is a null allele present in almost 40% of the population. Well, we can obtain respiratory tract cells from people who are both positive and negative for GSTM1. We can expose them to ozone in vitro and ask the question, does this polymorphism in vitro basically recapitulate what we know happens in vivo? And in fact, these are some experiments in vivo in which 19 people with the wild-type allele, 16 people with the null allele were exposed to ozone. And what you can see in the black lines is the people who are wild-type did not have very much of an inflammatory response to ozone. The people who are null definitely did. So clearly in vivo there's a difference in response between people who have the wild type and the null allele in terms of inflammation. If one then takes cells from those people, grows them in culture, the top panel actually shows GSTM1 activity, again showing that people who are null indeed have no GSTM1 activity. The people who are in wild type on the right have GSTM1 activity. And when they're exposed to ozone, you see again that the people who are the null are the people in vitro who are responding. So in this particular case, it appears that in vitro, we can begin to look at issues of susceptibility. We can now begin to ask questions about which toxicity pathways are activated. Are people who are GSTM1 simply more the same as wild type, or are there completely different pathways that are activated in this population of susceptible people? Another approach that we've looked at is disease susceptibility. Asthmatics are a big group of people, and they're exquisitely sensitive to a wide range of air pollutants. And one of the questions we wanted to ask was whether we could actually use in vitro technology to begin to look at mechanisms by which asthmatics respond. To do this, we obtained nasal epithelial cells, and note I'm talking about nasal, and I'll come back to that issue in a minute because I think it's important. From asthmatics, we took them immediately out of the nose. We isolated RNA so we could see what the asthma phenotype is from a person. We then placed them in culture, passaged them 3 times, and used microarray technology to compare passage 3, passage 2, passage 1, and immediately out of the nose. I'm going to mainly show you just the passage 3 because that's what's interesting. If you look at cells that are different between asthmatics and normal healthy people, one sees that about 13,000 genes are expressed in common between these 2 cell types, as you might expect. But the asthmatics have about 350 cells that are uniquely expressed in asthmatics. The normals have about 570 cells uniquely expressed by them. If one begins to look at the kind of genes, there are a number of genes that have been shown in the literature to be differentially expressed in asthmatics, and all of those genes that I've shown you here are indeed differentially expressed in our in vitro cells. So it appears that even though we've ripped this cell out of the body, removed it from its humoral and blood endpoints, taken away its underlying basement membrane, there still seems to be, after 3 passages, some aspect of these cells which still makes it an asthmatic cell. In fact, if you use principal component analysis, one can see that the asthmatic cells are clustering together and the normal healthy cells are clustering together. So we really think that this may represent a good model in vitro to look at susceptibility of diseases such as asthma. Well, are these cells more responsive to pollutants in vitro? And again, we've used microarray technology, but I don't have the data yet. We're about 3 or 4 weeks away from getting that. But what I do have is some data in which we've looked at specific markers. These are cells from asthmatics we did a few months ago. Again, we're looking at PM in this case, not ozone. And we exposed them to PM, and we looked at heme oxygenase as well as GM-CSF. And the normal healthy individuals are the green lines, the asthma are the red lines. And you can see that heme oxygenase in cells from asthmatics is upregulated following fine pollution exposure, not so in normal healthy people. And GM-CSF is not expressed in the asthmatic cells in either of the size fractions, whereas it is in the normal healthy cells. So it'll be interesting when we finish the microarray analysis of this to see exactly which pathways are activated. Once again, are asthmatic cells responding the same way, just more of the same, or they're actually different pathways that these asthmatic cells are using when they encounter toxicants. Well, most of the time today I've talked about bronchial epithelial cells. Those are not easy cells to obtain. We're fortunate that in our facility we do bronchoscopies 3 days a week, and so we have a ready supply. But it requires a board-certified bronchoscopist. It's an invasive procedure. It's not easy to do. On the other hand, obtaining cells from the nose takes less than a minute, doesn't require any medical expertise. It's non-invasive. There are publications out there showing it's been done in children, it's been done in all kinds of people. Easy to get these cells. The question is, how well do epithelial cells recapitulate what happens in the lung? Because if the answer is it does, then that opens the door to a lot of groups being able to do these types of susceptibility studies that I'm describing. So to answer that question from the same person, we obtained bronchial epithelial cells by bronchoscopy and nasal epithelial cells by brush scraping. Again, we cultured them, exposed them in this particular case to PM again, isolated RNA, and we've got the microarray for ozone, which is in the process of being analyzed now. But for the PM, what we showed was that we looked at 6 different endpoints, COX-2, IL-8, heme oxygenase, TNF, GM-CSF, and RANTES, all markers that we know are up in different degrees. And as you can see, for heme oxygenase, both the airway epithelial cells, which are in orange, as well as the nasal cells, which are in blue, are both up. We see that COX and IL-8 are not up in either one of them, nor is TNF. RANTES is also up in blue. These are cells taken from asthmatics. If you look at the normal cells, we see again that heme oxygenase is upregulated in both the nasal cells as well as the bronchial cells, and RANTES in this case is not. RANTES is typically made by asthmatics more so than normal healthy people, so we expected to see that type of comparison. So again, when we look at this and start seeing with the microarray, I think it'll be very instructive, but we're hoping that the answer will be that these nasal cells really are a good surrogate for the bronchial cells, which again will make it much easier for other people to do these types of experiments. In the final part of my talk, I want to remind you that we need to be looking at sometimes more than just one pathway. This represents all the kinds of things ozone does in people. It causes decreased lung function, if you look on the left, by activating C fibers, which then has an irritant response. Interacts with macrophages, which cause decreased host defense. It directly impacts mucociliary escalator function of epithelial cells. But we're focusing on what's in the pink. And part of what we need to do to develop really good models is we need to understand everything in those pink boxes from the time an ozone molecule hits the surface of the cell until the very bottom where it says inflammation. So far what I've been talking to you about has been near the bottom, the inflammatory mediators, the inflammation. But we're also working on some of these other boxes to provide data for the modelers so they can incorporate the upstream events. There's a large project underway, the Virtual Cardiopulmonary Project, which is looking at both the cellular level as well as the organ level. Again, trying to develop mathematical models that will take all of that and put it together into one model that can hopefully be used for a large number of pollutants, obviously not just ozone. This is an example of one of the upstream events that happens very quickly. Ozone is a powerful oxidant. It's been suspected for some time that it probably induces intracellular oxidative stress. This uses a combination of confocal microscopy as well as some of the newer cutting-edge recombinant green fluorescent proteins. And indeed, what you can see is in living cells, within 10 minutes after exposure to ozone, you see an increase in intracellular oxidative stress. We can show that that actually affects mitochondrial function. And it continues on for some time. So it's a very rapid event. What happens when intracellular stress occurs? Well, we know those ROS species interact with a very specific pathway. Intracellular signaling pathways usually involve phosphorylation of proteins, and the cell keeps a very tight control over that by a series of protein phosphotyrosines, which take the phosphates off, and protein kinase, which puts the phosphates on. Anything that interferes with that is going to cause dysregulation, and these ROS species, we've been able to show, actually inhibit the phosphotyrosines, which means the right side of that, the kinase pathway, now begins to run unchecked, induces phosphorylation of these compounds, which then affect downstream signaling pathways. What do I mean by downstream signaling pathways? Well, we all have our favorite charts. They're all complex. I'm going to focus on those three things circled in blue, although we've looked at many of the other pathways in there, the MEK and the ERK pathway. And this is studies showing that when we take these cells and we use antibodies that are specific for looking at phosphorylated MEK and ERK pathways exposed to ozone, we can see that ozone exposure causes an increase in the phosphorylated version of these, which interacts downstream on that NF-kappa B pathway that I talked to you about earlier. And I just want to finish by putting up, we all have our favorite adverse outcome pathway charts. If you ask 10 different people, you'll get 10 different charts. This happens to be one we like because we actually spend more time playing in those right-hand boxes than we do the left-hand boxes. For us, the public health and the individual responses are very important. So far, what I've been talking about today, however, has been more on the left-hand side, the molecular initiating events and the cellular responses. But we really think, for risk assessment purposes, we've got to bridge the gap between that left-hand box and that right-hand box of public health. Everything we do is geared towards doing that. And I think we all need to really be aware of that as we keep talking about this, that for risk assessment purposes, you've got to understand the individual responses and the public health consequences if you're really going to take in vitro data and use it for risk assessment. I'd be remiss if I didn't mention that the team that's been responsible for this, Kelly Duncan, Lisa Daly, are responsible for growing the cells, doing the experiments. Anna Fisher and James Crook are part of the group that's been doing the microarrays. Gary Hatch is doing the O18 ozone, James Samet and Sean McCullough, the upstream signaling pathways. And our group of modelers, Halal Burgoon, Steve Edwards, David Miller, Rory Connolly, and Hisham El-Masri. I'm not a robot, so I can't take 3 million questions, but I probably could take a few questions. And I tried to get us back on time.
Thank you, Bob. This was a wonderful presentation. Kind of getting back to our questions, I was wondering if with the example that you've started with, having cells from in vivo exposed individuals versus getting cells from, controls or pre-exposed and then treating them with a concentration and time course in vitro. So I understand you don't have that data yet, but once you get that data, what is your action? Should your hypothesis be confirmed, or should you find that there are actually differences in responses? So how would you then approach these two with respect to risk assessment or this in vivo, in vitro concordance?
Well, one of the things I should have mentioned, and it was remiss in saying, is that this project is not all about ozone. Ozone is a model pollutant, but we have exposed humans to a number of other compounds that we plan on using downstream to partly answer your question. We've exposed people to nitrogen oxide, sulfur dioxide, chlorine, volatile organic chemicals, wood smoke, diesel exhaust. So part of the goal is to ask the question, this is an oxidant, chlorine is an oxidant. We've already finished our in vivo exposures to chlorine. Are both compounds working by the same pathways? If what we learned with ozone is that applicable to chlorine, and if it is, what about compounds like phosgene that we can expose people to? Can we now say that we have a pathway for all oxidant pollutants? Can we now group them together? Same thing for VOCs. So this is just sort of proof of principle, if you will, that with ozone that this approach works, and we're hoping to apply it to a broader one. But the key to this, again, is the models. None of this works if we can't take and develop quantitative models to make these predictions, and that's what the virtual cardiopulmonary lung project is all about.
Right. Thank you.
Don Madison from Risk Sciences International. Great presentation. I really appreciate the focus on the inflammatory pathway, but would like some guidance on how you begin to think about chronic inflammatory states, because a lot of what you described really looks at sort of acute response of the inflammatory pathways. You're getting at it a little bit with the hypersensitive individuals, potentially those with asthma, but some thoughts about integrating this more chronic inflammatory state into the risk or hazard characterization paradigm.
Yeah, that's a good question, and obviously one of the limitations on in vitro approaches is it's very difficult to do chronic exposures in vitro. On the other hand, as you mentioned, one can take cells from people who have diseases of chronic inflammation like asthma and ask how they respond, which we've done. But if you're asking what can we tell from these experiments about someone who spends their lifetime in LA exposed to super high levels of ozone, the answer is maybe not too much, although I can tell you we can do more than acute exposures. One of the interesting hallmarks of in vivo ozone exposure is if you expose someone to ozone one day, they have an inflammatory response. If you expose someone to ozone 5 days in a row, they have nothing. It was like they never saw ozone, this so-called adaptation. We've been able to replicate that in vitro, showing that if we expose cells in vitro 5 days in a row, that this adaptation response occurs. So we can begin to look at not chronic in the sense of years, but we can, these epithelial cells live for months. So we can do long-term exposures, and we're in the process right now doing some exposures every day for 30 days to try to get at at the interface of what you're asking. But it's not an easy question, and I think for high-throughput screening, it's not likely going to be readily answered. But in some cases like this, I think we could start maybe approaching it.
Richard?
So I think you may have answered my question, Richard Dennison, Environmental Defense Fund, but it has to do with how all the time course data you showed for the in vitro system is time post-exposure, is that right? But you're talking now about exposure on a daily basis. Does your system allow for continuous exposure of cells and being able to better mimic, I suppose, what we would be envisioning human exposure to be? And could you describe, even in the data that you did provide, Bob, what is the exposure regimen? What length of time are the cells exposed, and so forth?
So we could do continuous exposure, obviously not for infinity, but for hours.
Yeah.
But what we were trying to do is tether the in vitro exposures to the in vivo exposures. If you look at the hundreds and hundreds of in vivo ozone exposure papers, the typical exposure is for 2 hours. That's sort of the gold standard.
Okay.
So that was what our in vivo studies are, and therefore for in vitro, we tried to also do a 2-hour exposure. What we did alter, though, was the dose delivered as well as the time course for in vitro, which is much harder to do in humans just because of the cost and time involved. But 2 hours is for both in vitro and in vivo is what most people are using.
Okay, thanks.
Bob, let me ask one more question from the ones that are up here. So your in vitro design with concentration response kind of implies dose response. And do you know, I mean, obviously this is, one of the information pieces or knowledge that you'll get once you complete your studies, but are you aware of any studies right now that do, concentration response comparison in vivo to in vitro? And, should we go to exposures in which we will not have in vivo data? Are we comfortable about, some sort of a scaling factor? I'm hesitating to call it an uncertainty factor, but, are we there with some information, or can we get the information to potentially start understanding what is the relationship? Is there just, similarity in the slope, but it's just shifted, or is it just on top of each other?
Yeah, that's a good question. And I'm reminded of the previous talks this afternoon that were emphasizing the issue of dosimetry. One of our take-home messages is that the concentration really matters. We know different toxicity pathways are activated at the higher concentrations than at the lower. We know in vivo that's true. Different endpoints are showing up at higher concentrations and lower endpoints. So I think in vitro, you've really got to try to do something to get at that notion that one dose is not going to tell you the whole story. You've got to have some kind of dose response, and you've got to have some confidence that your dose response is bracketing in vivo exposures. If we had done all of our in vitro exposures at the highest dose, that would not have told us what was happening to cells in vivo, because we know from the 018 that the 1 ppm is not really what the cells see in vivo. So I think it's not easy, but I think if we're going to try to get the right answer, we're going to have to try to take those things into account for risk assessment purposes.
But you're not aware of any data that would allow us to start thinking about this, scaling?
Not with this technology.
Okay.
There were a lot of studies done in the 90s looking at this, looking at specific markers of inflammation, but in terms of using this newer approaches of proteomics, omics, I'm not aware of any. No.
Suri Wulimari from USCP, NCR, Washington. That's a wonderful presentation. I mean, very good work it is you have been doing. I have one question in terms of you mentioned about the age of the cells, which is very important for the airway epithelial cells, right? So you mean it's like the culturing, like how many passages or something like that?
What I was referring to is that most in vitro experiments take cells and put them on plastic and use them that way. And for a lot of cells, that may be fine. If you're a liver cell, that may be how you are in the body and you're happy. But these airway epithelial cells in the body like to be exposed to air on one side. And when they do, they differentiate. And it takes them about 21 days in culture to differentiate fully, to make that mucin, to make those cilia, to have those hundreds and hundreds of genes whose now expression is changed from what it was. So when I referred to age, it was the time it took that differentiation process to take place in culture to where that cell now looks much more like what it would look like in a human body.
I have a hypothetical question. These cells, these are from either the asthmatics or the healthy volunteers, right? Like you're getting them. But does the age of those individuals affect anything in terms of the results?
That's a very good question. We have not addressed that in vitro. We certainly have addressed it in vivo, and age is definitely a susceptibility factor in vivo. It's one of the issues that we would really like to address, and that's one reason we went to nasal cells. It's not easy to do bronchoscopy on older individuals because of the risk factor, but now that if we can start using nasal cells, we'd like to know, are children more responsive, are older people more responsive? You can look at a lot of issues with nasal cells all of a sudden, because those are all potential susceptibility issues that I think need to be taken into account when we do these in vitro techniques.
Thank you, sir.
Let's take the last question from the microphone.
Thank you. Joyce Suji, Exponent. Thank you for that really nice talk, and that's a really unique system, and it's really going to help to move us forward. And one thing I had in mind to ask you was, you mentioned that there are very few data, less than 2 hours, and I've seen that is true. In fact, the shortest data, even in animal systems, is maybe 20 minutes to try to elucidate that concentration times time relationship for short-term ozone exposures. And for air pollution, that's not as big of an issue. But for people that are using ozone-based sterilizers in hospital settings or in the food industry, it is a real situation. You've got to figure out how much can you let somebody have exposure to in a short term. How do you design your equipment so you're not overexposing people? Do you think you could use your system to get a better concentration times time relationship for very short-term exposures, less than 20 minutes, say?
Well, it depends, again, on the endpoint you're looking at. We have looked at shorter exposures. I didn't report them here because it takes about 2 hours of exposure, both in vivo and in vitro, before we start seeing effects. But that doesn't mean that that's a downstream effect. As you saw with the intracellular oxidative stress, less than 10 minutes of exposure already caused intracellular oxidative stress, which was already beginning to affect mitochondrial function. It's just that that function didn't represent itself as a downstream overt inflammatory process. But one of the issues I think that's important when we talk about that, and this gets to the question that Isla had asked earlier this afternoon, and that is threshold effects. You're basically asking what is the minimum threshold that we can safely expose people to ozone. And I think this is an issue with toxicology that becomes critical, whether you're talking about individual threshold effects or population threshold effects. Looking at my favorite pollutant, ambient air pollution particles, I can tell you with a lot of precision what the threshold is for individuals and for cells, and I can tell you if I go below that threshold, I see nothing. On the other hand, epidemiology studies, over 150 of them, say there is no threshold for PM. It goes down to 0. And that's because there's someone out there who's old enough, sick enough, that even a low level that's below my threshold in normal, healthy people is going to push them over the edge. So I think it really depends whether you're talking about population thresholds or individual. And I think sometimes when we talk about in vitro, since most of our cells are from healthy individuals, and we say, this is a threshold, well, it's a threshold for that cell and that person. It may not be the threshold for that 80-year-old person with severe cardiovascular disease or that severe asthmatic. So I think we need to be aware that not everything is created equal when it comes to threshold effects.
Well, thank you. And let's give Bob another round of applause.